Hereby, I open this academic ceremony in which Sultan Qassem Mashnafi will defend the academic thesis, Human Cholesterol Metabolism, Effects of Aerobic Exercise Training, Nutrition and Inflammation. May I invite you to present a summary of your studies and the conclusions of your thesis.
Thank you. Dear Prorector, members of Corona, my supervisor, dear my family online, friends and colleagues, thank you for being here today. Within the next 15 minutes, I will give you a summary of my studies and a conclusion of my thesis entitled Human Cholesterol Metabolism, Effect of Aerobic Exercise Training, Nutrition and Inflammation. Cardiovascular disease is a major cause of deaths and diseases worldwide, in which atherosclerosis is an underlying process for the development of CVD. Atherosclerosis is an accumulation in cholesterol within vessel wall, and among several factors for developing atherosclerosis, such as high blood pressure and high blood cholesterol, high serum LDL cholesterol is a major risk factor. And this also also LDL cholesterol accumulates in vessels wall, and evidence in cholesterol metabolism has suggested that high intestinal cholesterol absorption is atherogenic. Cholesterol metabolism is maintained by the interplay between intestinal cholesterol absorption, endogenous cholesterol synthesis, bile acid excretion, and synthesis. However, a measurement for these processes require isotope tracers, and therefore they are expensive, laborious, complex, and not suitable for larger intervention studies. Therefore, an alternative approach using non-cholesterol using circulating levels of non-cholesterol has been developed. Non-cholesterol have a similar structure to cholesterol, and their plasma concentration are widely used for measuring cholesterol absorption and synthesis. Circulating level of cambisterol, cytosterol, and cholestanol are reflecting cholesterol absorption, while those of lathosterol, desmosterol are reflecting endogenous cholesterol synthesis. These sterol are transported in the blood by lipoproteins, so their concentration are standardized for cholesterol concentrations. First, we perform a systematic review to observe the association between the markers of cholesterol absorption and synthesis with different metabolic conditions. I have collected all relevant literature, analyzed the markers for cholesterol absorption and synthesis, and compared them to the controls in different metabolic conditions. We have selected 94 studies and clustered them based on the metabolic conditions. And the clusters were as follows: obesity, type 1 and type 2 diabetes mellitus, genetic and non-genetic hyperlipidemia, metabolic syndrome, cardiovascular disease, intestine, liver, and kidney diseases. The result showed that subjects with obesity, type 2 diabetes, genetic hyperlipidemia, metabolic syndrome, intestine and liver diseases have lower cholesterol absorption and higher cholesterol synthesis compared to the controls, while those subjects with type 1 diabetes, non-genetic hyperlipidemia, cardiovascular disease, and kidney disease have higher cholesterol absorption and lower cholesterol synthesis as compared to the controls. From this systematic review, we conclude that there was a clear inverse association between non-cholesterol steroid biomarkers for cholesterol absorption and synthesis, and the results of this systematic review suggest distinctive pattern for cholesterol absorption or cholesterol synthesis for different metabolic conditions. Based on the result from the systematic review, we saw that subjects with overweight or obesity are characterized by low cholesterol absorption and high cholesterol synthesis as compared to lean controls. And based on that, we addressed the following question in this population. What about the effect of exercise in cholesterol metabolism? What about the effect of weight loss in cholesterol metabolism? Does this profile relate to the chronic inflammatory status? Therefore, we did 3 studies to see the effect of aerobic exercise training, diet-induced weight loss, and systemic inflammation in the characteristic of cholesterol metabolism. First, we investigated the effect of aerobic exercise training in cholesterol absorption and synthesis in overweight or obese men. In this randomized control crossover trial, 17 obese men were randomized to start either with exercise or control interventions for 8 weeks. Men in exercise intervention, they performed a progressive aerobic training exercise on the stationary bicycle 3 times a week for 15 minutes, while those men in control intervention were asked to maintain their physical activity levels. After a washout period of 12 weeks, subjects switched interventions. Blood samples were collected at baseline, week 4 and week 8. And the result showed that aerobic exercise training for 8 weeks didn't significantly change the cholesterol standardized level for cholesterol absorption and synthesis markers compared with the control intervention. Next, we studied the effect of diet-induced weight loss in cholesterol and synthesis in cholesterol absorption and synthesis in abdominally obese men. For 54 men with abdominal obesity were recruited in a randomized control to see the effect of weight loss intervention. Besides, 25 men with a normal weight were included as a reference group. Obese men were randomized either into a weight loss group or no weight loss group. Men in weight loss group consume a very low caloric diet for 4 to 5 weeks to achieve a waist circumference below 102 centimeters. Then they consumed a mixed solid diet for 1 to 2 weeks, followed immediately by a 2-week weight maintenance period. Men in no weight loss group were asked to maintain their habitual dietary intake and physical activity levels. Blood samples were collected at baseline and after 8 weeks, and the data of markers for cholesterol absorption and synthesis were compared first between the abdominally obese men with a normal weight men and between the abdominally obese men before and after the weight loss intervention, and between the abdominally obese men after weight loss compared with the normal weight men. As we expected from the systematic review, obese at baseline have significantly lower cholesterol absorption and higher cholesterol synthesis as compared to normal weight subjects. In addition, weight loss significantly increases the cholesterol absorption marker cholestanol and decreases the cholesterol synthesis marker lathosterol. We didn't consider the changes in cambisterol, since cambisterol and the changes in cambisterol and cetosterol, since cambisterol and cetosterol are more abundant in plant-based diet, and due to the change in the habitual dietary intake after the weight loss intervention, during the weight loss intervention, their concentration are not truly reflecting the intestinal cholesterol absorption. In addition, there were no significant differences between obese men after weight loss and normal weight men for cholesterol absorption and synthesis markers. Again, as mentioned in the previous slide, we didn't consider the changes in the cambisterol and cetosterol due to the change in the habitual dietary intake during the weight loss intervention. Last, we investigate the effect of systemic inflammation induced by LPS infusion in cholesterol absorption and synthesis in normal weight men. LPS is a toxin derived from gram-negative bacteria, and it is a control model for inducing systemic inflammation response. Eight healthy young men were infused with LPS, and the blood samples were taken at baseline and at 24 hours after LPS infusion. LPS infusion significantly decreases the markers for cholesterol and bile acid synthesis, with no changing in all cholesterol absorption markers. To sum up, in our systematic review, we saw that subjects with overweight or obesity are characterized by lower cholesterol absorption and higher cholesterol synthesis as compared to lean controls. We saw that exercise intervention didn't significantly affect the cholesterol absorption and cholesterol synthesis, while weight loss increases the intestinal cholesterol absorption and decreases the cholesterol synthesis, and it causes a normalization in characteristic of cholesterol metabolism compared to normal weight men. Finally, we saw that inflammation induced by LPS decreases the synthesis in normal weight men, and therefore inflammation most likely doesn't explain the high synthesis seen in subjects with overweight or obesity. In overall conclusion, specific patterns for cholesterol absorption and cholesterol synthesis seem associated with different diseases, and these patterns can be modified by weight loss and the presence of inflammation. I would like to thank you for your attention. Now I will give the floor back to the Pro-Doctor.
Thank you very much for your clear presentation. We will now start with the opposition. And the opposition will be opened by Professor Jonkers, Professor of Intestinal Health at Maastricht University.
Thank you very much. Dear candidate, I would like to congratulate you with your very interesting thesis. I did read it with a lot of pleasure. I would also like to involve your supervisory team in my congratulations. I think it's really a very relevant topic to study cholesterol metabolism, especially in this era of increasing obesity and the associated comorbidities. But I would like to discuss a little bit further with you how we can use those differences between absorption and synthesis in the clinic, and we'll do that based on chapter 2. And we would like to ask one of your parents to read proposition 1.
Many metabolic disorders are associated with either a changed cholesterol absorption or a changed cholesterol synthesis, as reflected by cholesterol standardized plasma levels of campesterol, sitosterol, cholinesterol, desmosterol, and lethanesterol.
Sorry, I didn't want to stress you with all those difficult names. Thank you very much. And you also illustrated nicely in your presentation that you found distinctive profiles. And I'm not a total expert in this field, and I try to learn from all I did read. But I was also a bit puzzled that if you go to table 9 on page 45, where you made this nice overview of all the parts you found, and that you also mentioned it in your presentation, then you found this decreased cholesterol absorption with obesity and an increased synthesis. And I was actually a bit surprised, because I think overweight people have, and looking to their diets, generally have quite some high caloric intake, have a high food intake, maybe also cholesterol, and I was quite surprised that you found a low cholesterol absorption in those patients. Maybe to start with that, were you surprised as well? Can you explain it to me?
Okay. Highly esteemed opponent, thank you for compliments and for your question as well. If I understand your question correctly, you are asking about, you are doubting about why we are seeing a lower cholesterol absorption in overweight subjects.
Yeah,
right?
I was just surprised. Maybe you can explain it to me.
Yeah, I think if we look really at this kind of characteristic, also we saw it in a patient with a metabolic syndrome as well, and with type 2 diabetes patient. So I think that the factor here is more related to the insulin sensitivity or insulin resistance. So I guess that overweight or obese subjects, actually they have this kind of characteristic, so underlying characteristic that could be related that why they have a lower cholesterol absorption despite if they have really high caloric diet.
Okay, so that it was not just BMI or obesity in those studies, but
also we saw it also in a patient with a metabolic syndrome, also in a patient with type 2 diabetes.
Okay,
so
because is that then also the reason, because if I then go to that same table and then you look to the cardiovascular diseases, then you saw kind of the opposite.
Yeah.
And people with cardiovascular disease also have BMI as a risk factor, so I was also surprised there that it was the opposite finding.
Yeah, that's what we call it. It's like kind of distinctive pattern. So even I understand that also CVD patient, they also it could be maybe overweight. It could be they have kind of type 2 diabetes.
Mm-hmm.
But I think that's what actually we're trying to see, why we see this kind of pattern in the CVD patient, but we didn't see it in the people with the type 2 diabetes. So this is like kind of the opposite.
But do I now hear you say a little bit in between the lines that because you said you have distinctive profiles with all those metabolic entities, which you nicely did discuss per chapter, that it's not so easy to say that it's a specific profile per entity, but those people have not only obesity, but also maybe insulin resistance, so it's not so easy to say that it's a profile per entity. Is that correct, or is it not so easy?
Sorry, can you please rephrase?
You say that the people, or if you talk about the cardiovascular disease, there's also obesity, or there's also, for example, insulin resistance or diabetes. So it's not so easy to make a conclusion about people with cardiovascular disease or obesity because it's a mixed population. Is that correct?
Yeah, but if we look at the result here, then we are not talking about causality here. Actually, we are talking about the association.
Mm-hmm.
So we base our conclusion based on the association between the metabolic, a bit different metabolic condition with the markers.
Okay.
So,
Okay. And if you then talk about this association, because for this cardiovascular disease, I saw that you had quite some studies, and there was a number of studies who found an increase in absorption, some find a decrease, and quite some did not find an effect.
yeah.
Can it also be that there were inconsistent findings between studies and that it's not so easy to have a firm conclusion?
Yes, yeah, true that we have this kind
Because I
of...
saw that there was quite some inconsistency sometimes between studies. How to interpret that?
Yeah, but honestly, like the confident result that also we're going to see not only in the CVD, we also see it in other, could be also seen in other metabolic conditions.
Mhm.
But with actually what we're trying to do here is trying to see is if, sorry, can you, forgive me, can you but just repeat one more time?
I will take it a bit differently, because I think it is very interesting and also shows that it's very complex. And I was wondering, should we maybe look more on a personalized level? Because if you then go, for example, to figure 3 on page 109, then you have a very nice figure. And then you see that there's quite also some variation between individuals. Should we maybe more look to differences between being a syntheser and absorber on the individual level instead of a disease level? What do you think about that?
Yeah,
so what's your opinion?
Yeah, but in my opinion, this is going to be like a primary step to start with. And then we can look at the variation that between the individual. But for example, even if we say that, for example, overweight subjects, they are sensitized more than being an absorber, that we can still see the variation that some of them, they still maybe they are higher absorber than more than the low absorbers. So you're going to see this kind of variation for sure. But this is like a primary step to see how we gonna choose the optimal targeting intervention for this kind of people, for like different metabolic conditions. Like for example, in the people with I would say hemodialysis patient, they are at high risk of CVD, and so when they give the first treatment for this kind of people actually is statin. So a statin actually is affecting the synthesis, but they didn't see really that benefit from using the statin. But when they switched to use ezetimibe, they found there is really a beneficial effect on the LDL. So that's actually the point with looking at trying to understand more the cholesterol metabolism in different metabolic conditions. So it's more personalized approach. Indeed, we're gonna see CVD patient, but if you look how the time, like how many studies that I included in here for the CVD, so we need to have just a general look. We can't look specifically in every study. So, but at least we can have like a general look in the...
No, and I agree totally that it's good to do it in steps and first have a general look and then look further into... Can I ask one? No? Okay, then I'm very happy to discuss that later with you, and thank you very much for the answer.
Thank you.
Yeah, I'm sorry. Time flies. The next opponent is Dr. Van der Pol. He's assistant professor intensive care medicine at Maastricht University and Maastricht University Medical Center.
Yes, thank you very much. Dear candidate, first of all, I would really like to congratulate you with this very nice piece of work. I read it with a lot of pleasure and also want to involve your promotion team in the congratulations. Before I ask you the question, I would like to ask again one of the paranyms to read a proposition with less complicated words. It's proposition 7.
The doctor of the future will give no medicine, but will involve patients in the proper use of food, fresh air, and exercise.
Yes, thank you very much. as a medical doctor, I'm always very interested in what findings of basic physiological research, as the work that you have done yourself, what that means for clinical practice and for our patients. What can we do to use this new acquired knowledge to improve the disease course and to improve the treatment of our patients? So I was very much triggered by chapter 5, where you have administered LPS to healthy volunteers as a model for systemic inflammation and sepsis, critical illness, which is the patient category I'm dealing with every day. And you made some observations about alterations in cholesterol metabolism and also in bile acid metabolism, but I missed a little bit kind of an elaboration about the clinical relevance of these findings and whether these findings are beneficial or detrimental. Is it something which we should treat, or is it something we should stimulate? So my first question is if you can elaborate a little bit on the meaning of these findings, and then my second question will be what will be the nutrition or the diet that I as a doctor should provide to these patients to ameliorate these, to use these processes that you have studied to improve the outcome of the patients. Or maybe should there, after all, a medicine that we should use?
Esteemed opponent, thank you for your compliments and for your question. With regard to the clinical relevance of the study that we did when investigating the effect of LBS induced inflammation, actually, in this study, I know as you can see that we started with a really small number of subjects, but we wanted to have kind of basic understanding on the behavior of the cholesterol metabolism during the inflammation, because this kind of a study is the first kind of study, because no one had done that before. So we wanted to see how like the behavior of the cholesterol metabolism during the inflammation. So we use this more controlled model as LPS model, so for inducing systematic inflammation. And I'm still, my feeling is that even if that there was kind of an infection that like that viral infection or bacterial infection, we would have similar results in the cholesterol metabolism. So for the patient, I would actually first would like to recommend a lifestyle intervention. So before moving, like by changing the diet, by exercising, I see how that's actually affect.
Okay, I understand that, but unfortunately, in most of the cases, at the moment that I engage them in the hospital, that's too late for that, and we are engaging an acute problem. And also, you have used an acute model of acute inflammation to study some changes in cholesterol bile acid metabolism. So I'm in the first place really curious whether the changes you see, do you think that is kind of an adaptive, protective mechanism which we should stimulate, or do you see the changes you observed as some kind of thing that is going wrong and is a pathological state that we should fight?
Well, I believe that if the like this kind of changes for acute time, it's like now like what we have seen. This is kind of a protective role of the body against the inflammation. So when you see, when we saw this decrease in synthesis and for cholesterol and also bile acid synthesis, this also when I read in the literature is kind of to accumulate more to regulate the cholesterol content inside the cell. So it's more to protect actually to the host. But I think you are talking more about the chronic condition for the patients.
No, I'm actually, as an intensive care doctor, I'm very interested in the acute situation. So actually, the model you have used is a model of a disease state that is, for me, very relevant, and it really is relevant for me because it's an acute things, and therefore I'm also curious about suggestions of you as an expert, how we can use these observations to treat an acute illness.
Okay, so we saw here the markers for cholesterol synthesis decrease after the LBS in this acute phase. So if we wanted to treat, or maybe to have a beneficial effect for the patient after the acute inflammation, then it's quite difficult to decide what we should do, because our result showed that didn't show any kind of we did the correlation between the baseline desmosterol and the inflammatory responses, and we found actually the opposite.
Yeah.
So but when the studies like in human cell studies, they found something different. So so I think it's quite difficult to guide which treatment or which should we do in this in clinical relevance for the patient.
Maybe I'm a little bit too impatient, because you say this is the first in man study of this mechanism, and I'm already asking you how to use this in patient, and what nutrition or what drug I should give my patients. What do you think will be the most logical next step to finally in 5 or 10 years come to the situation where you can answer me the question, well, what to do, what I have to do as a doctor? What will be the next step to reach that goal?
I think we need. We draw kind of like a background about how the behavior of systematic review during the acute phase, and we maybe we need to look at what actually the behavior during the chronic phase as well. So at least if you combined those both results, then at least at that time or at that point, we would have more knowledge about how to deal with a patient with acute inflammation. So otherwise, I think it's quite difficult from here. I just to say what is the best treatment option for the patient with acute phase of inflammation.
Yeah. Is there time for another question?
Very short, and a very short answer.
Well, then I'll keep it for afterwards, because it's a long question I have in mind.
Okay. The opposition will be continued by Professor Gelijnse. Professor Gelijnse is Professor of Nutrition and Cardiovascular Disease at Wageningen University. I'm happy that you can be here. The floor is yours.
Thank you, Chairman, and respected candidates. Congratulations to you and your supervisors for this great accomplishment. I've read your thesis with great interest during the Christmas holidays. And now that the new year has started, I thought it might be good maybe for me to lose some weight. So I was especially interested in your Chapter 4, which is a weight loss study. Could you maybe tell what benefit I could have when you look at Table 1, if I would be in that intervention group with the caloric restriction? What would be the benefit for me?
Highly esteemed opponent, thank you for your compliments, thank you for your question. What do you mean by the benefit for us?
Well, if I would do the thing that these men were asked to do. So I think they were prescribed low caloric diets, and you measured several outcomes. And did it benefit their health, and in what way?
Yeah, okay. I think, here we can see like kind of lifestyle intervention. So when patient actually they are following a calorie-restricted diet for weight loss, and we saw indeed a benefit in terms of the metabolic profile, also in terms of the association between the cholesterol absorption and synthesis, because before the weight loss they were more with a more synthesizer, and now they are more absorber. So I think from this, the benefit is more here is more how I would say, it's like when you lose weight, then that's, as we can see here, that we saw kind of shift in the characteristic of cholesterol metabolism. So that means.
So it's better to be an absorber.
Yeah, if you could, but they become more absorbers. So that is in terms of the insulin sensitivity, they have. I think Matt does more related. So that's they have
I was
more
hoping that maybe both the absorption would be influenced, but also less synthesis. That I would have both benefits.
Exactly. So because both the synthesis and absorbers they have kind of reciprocal relationship. So if the one factor affects, we would assume to see an effect also on the other factor. So this is beneficial in terms like they have more, they don't synthesize more cholesterol compared to the obese people. So after the weight loss, for sure, this is more beneficial, not only in terms of the cholesterol metabolism, also in terms of other metabolic profile.
So it's a good thing to do.
Yes.
Huh? So, but what do I have to do then? Do I have to take these ModiFast products?
I think you can. I think can also can lose weight by other interventions like. Is not only by isocaloric, it's also that by doing exercise and losing more weight. So it's
so when to what intervention do you actually attribute these findings? Is it the weight loss per se? Is it the change in body composition? Or is it the change in dietary intake that causes these effects?
Okay, so to answer this question, based on the changes that we saw, based on the association between the BMI with the visceral fat and also with intrahepatic lipid, these two factors actually are one of the important factors that related to the CVD risk. So when we did the mediation, we wanted to answer that question. We want to see how much the effect actually.
Well, actually, that's not an effect, because you did the mediation analysis on the cross-sectional data.
Yes, true.
So that's not an intervention study, and you cannot be sure about the causality because it's a cross-sectional design. I'm now sticking to the intervention, so to the treatment effects. So are these treatment effects due to the actual weight loss or the change in body composition, or by the fact that they consumed another type of diet? So what actually did they eat? Did they consume phytosterols? What actually is in the ModiFast? Were the fiber levels similar in the intervention and control subjects? What do you know about what they actually consumed?
So honestly, I don't have that much idea about the formula they used. But if we look at the content, I think it was more like high protein, low fat formula. So and also I'm expecting that also they were allowed also to consume vegetables.
So apart from ModiFast, they could also pick some other foods.
Yeah, I can have vegetables also or fruits. So, but
and what about coffee and tea? Could they also consume coffee and tea and other beverages?
Coffee and tea, I think the coffee and tea. I'm not, to be honest. If I don't have really these two details information because this study wasn't primarily done by me. So a co-author actually done this study, but. The idea that I have about the dietary food, the dietary consumption, they use this kind of formula beside to the
But you
vegetable.
don't know what exactly they were eating. You don't know the exact amount of phytosterols.
No,
that
we
they-
didn't. So they
Okay.
didn't measure that the plantasterol
Okay.
concentration. No.
I have another question because I'm also interested in personalized dietary advice. So I'm leading a couple of intervention studies myself. And we are now moving in the direction of personalizing these diets to an individual's need. Do you think your knowledge about the differences in patterns for cholesterol absorption and synthesis, could that help in giving more personalized nutrition advice? Could you speculate on that?
Yeah, indeed. For example, if for a subject with high absorption, we can advise them to have kind of who they, I would say they are relatively absorber. We could advise them to consume diets that affect the intestinal absorption. For example, such as a plantasterol and plantastenols. So these kind of these are dietary products could for sure decrease the cholesterol absorption in those type of individual. So, for example, in overweight, if we want to, actually the risk marker is more the LDL. So if you want to lower the LDL, so we need to look also the phenotype of the participants. For example, if it's absorber, then we can use plantasterols, or we can use ezetimibe in addition to the plantasterol or plantastenols. And for those who are synthesizers, we can ask them to have more kind of drug intervention, like statins or bempedoic acids, for example, or this kind of intervention. I would try ask or give an advice for.
It's not easy. I'm also trying to translate it into daily practice, and also the table that former opponent talked about. Like for cardiovascular disease, the pattern may be different than for obesity and diabetes. So, and these are often clustered, these types of diseases. So it's, I think, some work to do to figure out who exactly should get what advice.
Yeah. So if I think this steroid could give us an indication, but analyzing this kind of steroid, non-cholesterol steroids, could give us an indication for what in advising the best intervention for either dietary or pharmacological for the participants or for the patients. And for example, in type 1 diabetes, they are insulin dependent, and we saw that they have higher cholesterol absorption, and the studies showed that when they are actually treated with statin, there is no much benefit on the lowering the LDL.
Oh, that's a bold
but when
statement.
we treat them with ezetimibe, they reach kind of the optimal LDL levels. So that could be.
I'm trying to avoid the prorector, but she's looking at me, so I give the floor back.
Thank you very much. The opposition will be continued by Dr. Trautwein. She is a visiting professor at the Christian Albrechts University in Kiel in Germany. Thank you for coming to Maastricht. The floor is yours.
Thank you. I didn't come from Kiel, just to be clear. This would have been a very long travel, but I came much closer. So, dear candidate, I also like to congratulate you on your thesis, which is a very nice piece of research, and I also congratulate your supervisors for this achievement and also for the nice publications, and especially the systematic review. I have read with great interest because cholesterol metabolism is also my research interest. Particularly, I want to focus on intestinal cholesterol absorption. You have shown it in your 50-minute presentation, and of course it's also written in chapter 2, where you say that a high intestinal cholesterol absorption is associated with a higher risk of cardiovascular disease compared to cholesterol synthesis. And could you say or tell us what the underlying mechanism for this could be? Why is high cholesterol absorption increasing risk, whereas you would expect that hepatic cholesterol synthesis also has such an effect?
Esteemed opponent, thank you for compliments and for the question. And if we look at this association between the higher intestinal cholesterol absorption in a CVD patient, well, I think we look here as association, not a causality. So that is make it more difficult to look at the underlying mechanism behind that. So, but if this information, sorry, could you repeat the question? I forgot what was your...
It's why is cholesterol absorption increasing CVD risk compared to cholesterol synthesis, and what's the underlying mechanism? You're right. I mean, this finding stems from observational studies, and observational studies like the Framingham study can only give us an association. Are you aware of any intervention studies that have shown that cholesterol absorption increases risk? Or if that's the case, the opposite would also be true. If you lower cholesterol absorption, the risk should go down. Yeah,
if we look here, when we use plantosterol to reflect more the cholesterol absorption, so it's difficult to distinguish these two things, plantosterol and absorption. So I think is quite, I would say that, because we saw kind of reciprocal relationship. So as you said, that higher and lower, but this kind of only based on the association, not the causality, and we want to see if this kind of distinctive pattern for the CVD patient, like for hemodialysis patient, they are higher risk of CVD, and when we're treated with ezetimibe, they reach better LDL lowering, but I don't.
So if there would be an intervention with ezetimibe, which would lower cholesterol absorption, one would expect to see a lower risk of cardiovascular disease compared to an intervention with statins, which would lower cholesterol synthesis. I mean, would that be the case? If such studies would exist, of course.
Yeah, but I think in the beginning, like first cases of cetoasterolemia, when in the bad first cases they discovered kind of cardiovascular disease with those patients, and in the beginning there was kind of argument maybe plantosterols that actually reflect in the intestinal cholesterol absorption here, they are more causing this cardiovascular disease events, but when but the time being now that nowadays we can see more people with cytosteremia who are healthy and don't have the really CVD events.
And I mean, cholesterol absorption markers, there are basically three. There are the plant sterols, cytosterol, campesterol, and you have the cholesterol product, cholestanol.
Yeah.
I was recently being asked, which one would you measure if you only have to measure one? Or would you actually say, no, one has to measure all the three? What would you answer?
Well, I think if you ask me to measure one, then, but I prefer to combine at least one, like if measure one, I would like to have a cholesterol, but honestly, I would like also to combine at least one of the plantosterol next to cholesterol. And also, if the studies that they looked in the CVD patient, they also looked at they specifically look at the cholesterol levels, because cholesterol is less influenced by the dietary change. So it's not really that much available in our diet.
And why would you measure, well, if one could only measure one, why would you choose cholestanol and not a plant sterol?
Yeah, because cholestanol is really limited in our diet, so it's normally is like formed from the cholesterol. So and normally cholesterol when reach the colon is actually more metabolized by the colon bacteria to non-absorber metabolites. So that's going to be excreted. So I would but for example, if we only depend on the plant sterols, that this kind of compounds is really to be influenced by our diet. So because they are more abundant in our diet.
Okay. And could you say something about the validity of these biomarkers? I mean, have they been validated to actually, relate to cholesterol absorption if you measure it via isotope techniques or tracer techniques?
Yeah, but those markers, do you mean for the cholesterol absorption markers, or just
the absorption markers?
The cholesterol absorption marker, yeah, they were really a positive correlation between these markers and the absolute measurement they were used for measuring the cholesterol absorption. For example, the cholesterol balance method, and also the isotape dole continuous feeding method. This is actually a method that really using tracers, and we are actually but the benefit that we're gonna get from the measuring these plant sterols is because you'll just only need a single blood sample, and then you can get an indication about the fractional rate of the clinical absorption, the intestinal absorption.
Yeah, I mean, I understand that the surrogate markers make it easier to measure, but is there a validated method for this? Or, I mean, can every lab basically say, oh, I'm going to measure plant sterols and use them as cholesterol absorption biomarkers?
Yeah, they are really reflecting the cholesterol absorption, but there are large variation in their concentration, and that's actually due to the variation in the method techniques that are actually by different labs they are using to measure the non-cholesterol sterols in general, and that makes it more difficult to work with the normal values for these sterols. And also, in addition, it's going to be difficult also to defining later on the absorber and synthesizer, because we really have this really large variation in the techniques they are using to measure these sterols. Yeah.
So I assume you have used a validated method in your study. I mean, knowing your supervisors, I would definitely assume so. Yeah. But why is it also important to always adjust for cholesterol and use the ratio?
Yeah, because
I mean, would it be not better to use the absolute concentrations?
I would, yeah. Well, I think since these sterols, they are really behave like cholesterol, and they actually transport in the blood by lipoproteins, and the content of the lipoproteins is there is a variation in content of cholesterol in the lipoproteins. So I think it's better to standardize it from the total cholesterol, just to eliminate this variation in cholesterol content in different lipoproteins. Yeah.
Okay. Do I have still time?
No.
Okay.
So we now start with the last opponent. That's Dr. Adam. She is assistant professor at Maastricht University.
Dear candidate, first I would like to congratulate you on your thesis. And of course I would like to include your team of promoters and your co-promoter in the congratulations. My expertise is not cholesterol metabolism, but I read your thesis with a lot of interest in the context of obesity, which I study. And therefore, I particularly have questions for you based on chapter 4 regarding the weight loss study. So in your overall introduction, it says that high intestinal cholesterol absorption is arthrogenic, and it's estimated that approximately 30% of the population is characterized by high intestinal cholesterol absorption. In your review and also in the intervention study, it suggests that obesity and higher BMI is associated with lower cholesterol absorption. And there I had a similar question, or I was similarly surprised than compared to your first opponent, because it seems counterintuitive to me that obesity goes along with a higher absorption. So since I'm interested in food intake regulation and obesity, we know that most aspects of food intake regulation ideally, at least in part, go along with homeostatic regulation and negative feedback mechanisms. So my question is, what you find or your observations in your participants with a lower absorption, could that be some sort of a protective mechanism in people with obesity? Could that be a reaction to the obesity rather than a primary pathological occurrence?
Esteemed opponent, thank you for your compliment and for your question. Do you mean after the weight loss? Do you mean for the obese after the weight loss, or?
No, in general.
In general?
Yes, before the weight loss even. Because even at baseline or in general, we find obesity, or that is what you describe in your thesis, that obesity goes along with a lower absorption, cholesterol absorption.
Yeah, so you are wondering why we see this kind of pattern of low absorption in obese?
Yes, and so my question is that some sort of a protection of the obese individual? Is that some sort of a feedback to higher circulating cholesterol in the blood?
Well, I think, yeah, indeed, that there is a reciprocal relationship between the cholesterol absorption and cholesterol synthesis. And that's when we see kind of an increase, then that will be faced by a commensurate decrease in the cholesterol synthesis. But if we look at like not only in the obesity, we look at other patient with, for example, the patient who are they don't have, or they have a deprived small intestine. So here we're gonna see like a kind of absence of the cholesterol absorption, because that part of the small, or by the way, a huge part of the small intestine is actually removed. And we still see also there is kind of a compensatory, because there was an increase in cholesterol synthesis. And but I can't say is more a protective thing. Actually, for example, in LBS study, is more a protective way of our human body against the inflammation. But we only saw the effect on the cholesterol synthesis. So that is quite difficult to say is a protective way or not. But we are looking more on the association. So we don't know if it could be there is another underlying factors behind this relationship between cholesterol absorption and cholesterol synthesis, and also more factor we should consider. But I can't say is a more protective or not protective for obese people to see that in obese people, this kind of pattern of lower cholesterol absorption, but could be more related to the insulin resistance. So because when we look at type 2 diabetes, they have also lower cholesterol absorption. So and this and metabolic syndrome, they have the same. So it could be something that I think the factor that connected all these together actually is more the insulin sensitivity. So yeah.
But in obesity, it was also observed that the binding of LDL to the LDL receptor is reduced. So could that have anything to do, or could that be responsible for a lower absorption of cholesterol? Is that related?
Yeah, honestly, there is no direct link between the cholesterol synthesis and the LDL receptors. Sometimes you have a decrease in the synthesis, but there is no effect on the LDL receptor. So I think it's quite difficult to judge or to say that it's actually because of the LDL receptor is decreased.
Okay.
So, but there is no direct link actually.
So what would your conclusion then be? What is metabolically more relevant? Would that be the low absorption or the high secretion in obese participants?
And but I think, since this kind of reciprocal, so I think it's more we should focus on two pathways, so not only once. So,
okay.
Yeah.
Okay. So you would look at them as separate entities.
Yes.
Okay.
Sultan Qassem Mashnafi, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and, of course, your defense. And I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. What's on the line? I know you know that fly by now, don't waste all your time. Cause I'll go the extra mile.
fine.
Sultan Qassem Mashnafi. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Platt is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Sultan Qassem Mashnafi, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree of certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university. So, Dr. Mashnafi, dear Sultan, congratulations with this achievement. I think you gave a very clear presentation and the defense went well. I think you can be proud of yourself, at least we are as a supervising team. But I will give the floor to Sabine because she was most closely working together with you, so she will give you the laudatio. But congratulations.
Thank you. So, well, dear Dr. Mashnafi, dear Sultan, of course I also want to congratulate you with your thesis, but I would also like to take this opportunity to review your time a bit as a PhD student at our department. So you started your PhD program 5 years ago, and indeed, as you told us today, our aim was to measure non-cholesterol levels and to link this to all types of interventions and conditions. Well, for this, you had to learn the GCFID technique to measure them, and you spent many hours learning all the details from Maurice. It was a huge undertaking, and in the end, you persevered and you were able to master this technique. So I really want to congratulate you with this, the huge amount of work that you did on this. As said, the common denominator in your PhD program were the sterol levels, and for your first publication, we also did perform this systematic review. So you assess them as biomarkers for synthesis and absorption, as you told us today. And it was actually quite nice of a start of your PhD because you really had to dive into the literature and thoroughly had to assess all the content to see whether it would fit our paper. You did this very well and it resulted in a nice publication, so congrats. We then had a minor setback because we wanted to measure samples from the Maastricht study. This was unfortunately postponed, so we had to come up with an alternative. So instead of doing this large GCFID study, we were fortunate enough to collaborate with Peter and with Jordi, and we could retrieve samples from their studies. So you assess absorption and synthesis after aerobic exercise from Jordi's trial, and from Peter's study, we were able to get the samples from the diet-induced weight loss trial. So, and both studies were eventually published in very nice journals, so well done. But you also had to learn this mediation analysis. So, for the ones who are not familiar with this, I'm not really an expert in statistics, but it's a really complicated technique. And together with Ronald, you were able to dive into this and really perform this meta-analysis or this mediation analysis for us in the weight loss trial. So well done to you. During the last part of your PhD, you worked on, again, 2 new topics, so systematic inflammation and liver function. And of course, for us, it was very nice that we were able to include so many topics in your thesis, but for you, it also meant that you had to get acquainted with a lot of different topics. So this requires really good adaptation skills, and that's something that you did very well throughout the publications that you did. You had to read so much different things every time, focusing on the steroid levels, but all with different topics, so well done. During your PhD, you were working hard, but it also took a little bit of time to get accustomed to our European way of working. There were, of course, there are small differences in our cultural background. And for example, it was clear in the beginning that it was difficult for you to ask for help, or maybe to say that you didn't understand something when we had a discussion, what would be the next steps of action. However, I hope, well, we resolved that really fast in the beginning, and I hope that you felt comfortable enough throughout your PhD to come to me for help or to ask Jochem and Ronald for help when you needed it. Another aspect that I really think that you improved during your PhD time are your presentation skills. So you presented several times at the Dutch Nutritional Science Days, and also you had a nice poster here at the EAS in Maastricht. And I think when you started, you were quite hesitant to do presentations, but it improved over the years. And I hope that you had that experience that you had in these 5 years, that they're helpful for you in your future career in that aspect. Then, as many of us can relate, the past few years during COVID-19 were not that easy, but I think they were especially difficult for you. The restriction to stay at home as much as possible, to work from home, to refrain from social activities really resulted in a lonely time for you. You were able to manage through this difficult time, and I really think that again indicates your perseverance throughout the last years. Then I've also gotten to know you as someone who loves to travel. Unfortunately, again, due to COVID, you were not able to travel as much as you would have liked to, but I hope that you still were able to enjoy the Netherlands during your stay. And I think that there's also one thing that you missed during your time here, and that was all the nice foods that you have at home. So ultimately, I think you found a nice restaurant in Belgium that served the grilled lamb meat that you liked so much. And that's something that you can look forward to now when going back. So all the nice foods from home again for you. So Sultan, I really want to thank you for a nice time. It was a pleasure having you in our group, not only as a PhD student, but also as a person. I'm happy we got to know each other, and I wish you and your family all the best and all good luck in your future career. I would like to leave it as a short review for now, so I wish you an unforgettable day, all the best to the future, and with this I'd like to give the floor back to the prorector.
Dear Dr. Sultan Qassem Mashnafi, also on behalf of Maastricht University, I would like to congratulate you with the degree you have acquired today. I really enjoyed being present at this PhD defense. I thought I knew something about cholesterol metabolism, but I learned a lot again today, as almost in every PhD defense. So it was quite enjoyable. I hope you enjoyed your defense as well. You seemed a little bit nervous in the beginning, but later on it got better, and I hope you can look back to this ceremony with a lot of satisfaction. I think you deserve it. I would like to congratulate your promotion team, and I would like to thank all members from the assessment committee today for being present here, especially our guests from outside, Professor Gelijnse and Dr. Trautwein. Thank you very much for taking the time to come here, which is in our Zoom time quite special that everyone is really live and present. So thank you very much for that. And well, with that, I'd like to wish you a very nice rest of the day. I hope you have some time to celebrate with some friends, or maybe I understand your family is not here, but maybe they can celebrate in Saudi, and also have a nice time and be very proud of a family member that has a degree from Maastricht University. So with this, I close this academic ceremony, and we are going to take a picture outside on the stairs and congratulate, and I'd like to ask